So we're going to look at action potentials and how they're formed and go through the processes that are involved. So if we start by just looking at the graph, then that will be the easiest way to explain it. So here is our graph. We have time against potential difference. Now, when we're talking about potential difference, we are talking about comparing the inside and the outside of the neuron. So it's measured in millivolts, it's very small, and it starts at what we call minus 70. And then once you have a stimulus, what we notice is that first of all, it goes up relatively slowly to approximately minus 55, and then it shoots up quickly to plus 40, and then comes back down again, slightly overshoots to minus 70 and returns to that level. Okay, so what we actually need to think about is we need to think about what is happening at each stage along this process. So if we start with minus 70 here, minus 70 is called the resting potential. Go through and label the parts first. You then stimulate and it reaches minus 55, which is called the threshold potential. And we'll see that the potential difference must reach minus 55 if you are then going to go up and generate the action potential of plus 40. And this process of going from minus 55 to plus 40 is called depolarization. And then going from plus 40 back down is called repolarization. So you are repolarizing. You reach a point where it's below minus 70, which is called hyperpolarization. And then you go back up to the resting potential again over here. So we'll just label that as the resting potential. So if we have a look at what is happening here. So let's just think about a neuron first of all. So we know that a neuron is made up of a phospholipid bilayer. So here is our neuron. Here's one membrane. Here would be the other, but I'm actually going to draw it in as a phospholipid bilayer. So here's the inside, and here is the outside of our membrane. So we have a phospholipid bilayer because all membranes are made of phospholipid bilayers. And within that membrane, we have various proteins that are important. So we're going to do the first one over here, which is a common carrier protein called the sodium potassium pump. We'll put some more phospholipids in, forming a bilayer. We've then got some channel proteins. We'll put a few of them in. These are sodium channel proteins, and they can exist in one of two types. They can be non-voltage gated, or they can be voltage gated. There are two different types, as we shall see. And then we also have some potassium channels, which I'm going to leave some of them as open. We're going to need them to be open. And we also have some that are closed. Just put in my phospholipids between them. So, and again, some of these are voltage gated. That means they will open in response to certain voltages. But these are potassium channels. And some of them, like these two over here, are non-voltage gated. Okay, so let's think about what happens in the formation of our resting potential. Okay, so that's what we're looking at here. We're looking at our resting potential and how we get there. So the first thing that's going to happen in the formation of our resting potential is that the sodium-potassium pump will actively transport 3 sodium ions out for every 2 potassium ions that come in. Now, the other thing you will notice is that my sodium ion channels are closed. Some of my potassium ion channels are open. So because it's more permeable to the potassium ions, some of these potassium ions which diffused in will diffuse, which were transported in by active transport, will now diffuse back out. So how do we describe that? Let's put it in words. So number one, okay, the sodium potassium pump actively transports 3 sodium ions out for 2 potassium ions in. You'll notice I use the word ions. It's really important that you refer to ions and not to sodium and potassium, otherwise you will lose marks. And then we go on to say, however, as the membrane is more permeable to potassium ions than sodium ions, some of the potassium ions will move back out by facilitated diffusion. Note, I'm saying what type of diffusion it is, not just diffusion. And this creates a potential difference of minus 70 millivolts. Okay, and that is what is called the resting potential. So that's the first stage that's going to occur. So if we go back up to the graph, that is what is occurring in this particular process over here. This formation of this resting potential. Okay, we then get a stimulus. So we need to think, well, what happens when that stimulus comes about? So if we go back to my membrane over here, I'm going to remove my ions. Remove my arrows for a minute. Okay, so what we're going to find is that once we've reached resting potential, then these potassium ion channels will close. However, when you've got a stimulus, what will happen is that the non-voltage gated, so let's assume this is our non-voltage gated, the non-voltage gated sodium channels will open, which means that the sodium ions, which were pumped out before, will now move back in by diffusion. And that will bring the inside of the membrane to about minus 55 millivolts. And once it reaches minus 55 millivolts, that is enough to then open up my voltage gated sodium channels, more sodium ions diffuse in, and then you go from minus 55 to plus 40, which is your action potential. Let's put that on the graph. Okay, so if we say what's happening here, and then what's happening here. So a little bit smaller, change its color. So when a stimulus arrives, we'll look at stimuluses later and how they actually cause these sodium channels to open. Sodium channels open and sodium ions diffuse in to reach the threshold potential. Because I've mentioned channels, I don't need to say move in by facilitated diffusion, because I've already talked about the fact that it's moving through channels. Okay, so that's that one. And then over here, we can say when the threshold is reached, voltage gated sodium ion channels open. Sodium ions fuse in and cause depolarization. We must learn to use the terms. An action potential of plus 40 millivolts is reached. Okay, so we've now got our depolarization process. So we then reach our action potential, and then we're going to have to try and get the potential difference back down to minus 70. So what's going to happen next is that when we reach the action potential, this time the sodium and ion channels will close, and the voltage gated potassium ion channels open. Potassium ions diffuse out, because remember they were pumped in the formation of the resting potential, causing repolarization. Okay. However, too many potassium ions leave, causing hyperpolarization. So we've actually got too much leaving, and that's where our problem becomes. Too many leave. Okay, so if we go back to our picture here, what happens is once we've reached plus 40, these channels will now close over there like that. Can no longer transport anything in. So get rid of that. Our potassium channels, however, will open, and our potassium ions will now move out. And that takes it from plus 40 to about minus 85 millivolts or lower, which is the hyperpolarization. And then last thing you're going to have to do is over here, which is restore the resting potential. And you do that in exactly the same way as you made the resting potential in the first place. Now, bear in mind, however, that if you don't reach the threshold, which can happen, if not enough sodium ions come in the first place, and we don't quite reach the threshold, then it will just go back. Okay, we call that the all or nothing response. The all or nothing response means that you either reach the threshold and go to action potential, or you don't generate an impulse at all. It's one or the other.